It's my pleasure to introduce Professor Jessica Metcalf. Professor Jessica is a biologist by training with a bachelor degree from Oxford, followed by a PhD from Imperial College London, both in the UK. After a period as a postdoc in many different institutions, which is amazing, she was in Max Planck in Germany, University of Pennsylvania, Duke, and Princeton in the US. She became a research fellow at Oxford University. And currently, she's associate professor in ecology, evolution, public affairs at Princeton University. In her work, she's interested in characterizing the landscape of immunity to support public health and developing a framework for understanding the evolution of immune function. So for the disease people, this is your week, guys. And we also have here Benny Rice. It's also my pleasure to introduce Dr. Benny Rice, who is currently a postdoc at the Department of Ecology and Evolutionary Biology at Princeton in the US. And he's a microbiologist by training with a bachelor degree from Arizona State University in the US and a PhD from Harvard University, also in the US. Currently, he works on genomic and serological analysis of infectious diseases across a wide diversity of community and ecosystem settings in Madagascar, which is great. Hey guys, it's your chance. And he also wants to understand how the variation in commonly circulating viruses, including coronaviruses, is shaped by interactions with immune responses, combine field-based epidemiological studies with lab and theoretical approaches. So help me to welcome these two.
Thank you so much. Can you all hear me? Yes. Okay, great. Well, it's a pleasure. I think, could we start with a round of introductions, just so as we get your names and know your background? Would that work? Just so as I have a sense? Yes.
Okay, hello. My name is Denise. I'm from Argentina. I'm a physicist by training, and I'm about to start my PhD talking about the role of heterogeneities in epidemics in general, like COVID, Zika, malaria, and I did my master's also researching COVID dynamics.
Fantastic.
So I'm Thales. I have a biomedical science background. I study proteins isolated from snake venoms, also the biological effects against Toxoplasma gondii infections, multibacterial resistant biofilm formation related strains, and other aspects.
Fantastic.
Hi, I'm Camila. I come from Colombia. I'm a biomedical engineer. I just started my master's last semester, and I usually work with mathematical models in dengue.
Fantastic.
Hello, my name is Julieta. I'm from Colombia as well. I'm a physicist, and I have worked a little bit with molecular dynamic simulations of proteins.
Hi, my name is João Carlos. I'm from Brazil, and I am a biomedicine undergrad student, and I work with molecular biology inside of immunology.
Hi, my name is Abel. I'm from Brazil as well, and I'm a biologist. Right now, I started my master's degree with animal evolution and speciation.
Hi, my name is Juan. I'm from Colombia. I am a biomedical engineer, and I am doing my master in biomedical engineer, specifically in mathematical modeling in COVID.
It's popular.
Hello, my name is Adenilson. I'm from Brazil. I studied mathematics before, and I'm studying biotechnology, and I have a lot of interests.
Hi, my name is Amanda. I'm a biomolecular physicist, and I'm studying applied mathematics. I'm like a lot of modeling.
Hi, my name is Sebastian. I'm from Colombia. I'm a biologist. I like evolutionary biology and information processing in nature.
Hi, I'm Tawane. I am an oceanographer.
I finished my master's degree last year, and I'm looking for a PhD, and my interest in animal movement and movement ecology.
Hi, my name is David. I'm from Colombia. I work with malaria and population genetics modeling, and I'm a physicist and a biomedical engineer by training. I'm finishing my master's degree in biomedical engineering too.
Hi, my name is Erika. I'm from Mexico, and I just finished my undergraduate in genomic sciences. And my main interests are systems biology and work with data, any kind of data.
Hi, my name is Ruth. I'm from Mexico as well. I'm a mathematician. I have finished my master's degree, and I'm interested in epidemiology. I have worked in COVID.
Hi, my name is João. I'm here from Brazil. My background is in biology, and I just finished my master's in ecology.
Hi, my name is Benjamin. I am a physics student here at ICTP. I work with statistical physics and applications to movement ecology. Yeah, things like that.
Hi, my name is Kawe. I am currently doing my master's degree in mathematics, but also I have my undergraduate degree physics and molecular biology.
Fantastic.
My name is Thales. I'm from Brazil too. I'm a biologist, and I'm the end of my master's now in botany, and I work with plant evolution.
Hello, my name is Julia. I'm from Brazil. I'm doing my undergraduate in biology, and I'm mainly interested in ecology.
Oh, my name is Nicole. I am also an undergraduate student in biology, and I am developing a project in ecology and competition.
Hi, my name is Mateus. I'm from Brazil too. I'm a physicist, and I'm currently starting my master in neurodynamics, critical states, and this kind of things.
My name is Amanda, and I'm from Brazil, and I made my bachelor in ecology and finished my master's in ecology too.
Hi, my name is Gislaine. I am a veterinarian, and currently I'm pursuing a master's degree in bioscience and biotechnology, mainly with visceral leishmaniasis in different aspects.
Hello, my name is Cristiana, and I finished my undergrad in applied mathematics, and now I'm looking for a master's program.
Fantastic. Well, thank you so much. You are a diverse group. so I'm going to try and cover this in a way that, that doesn't seem too easy for some of you or too hard for others, but I'll probably get it wrong at times. So forgive us. And I think we've tried to design this so there's lots of options and directions you can go in that aren't covered in the classroom, and that we can just be talked to about. So hopefully that will create some flexibility to join your huge flexibility. Course logistics. So we have so there's a text that sort of tries to cover a vast array of epidemiology, which I've been working on for a while. So it probably has many mistakes. If you find any, please share them with me. But that should give you a context for some of the things we'll talk about. There's also a set of little snippets of R code that go with many of the lectures. There'll be references at the bottom. The Dropbox folder is there. I think we're also going to email it around, or we have emailed it around?
I sent it on Slack. There's a Dropbox folder linked on Slack that has this PDF, and you can get those links from there. And do ping Slack if the links are not working, and I'll try to fix that in real time. So raise your hand later or bother me later if it's not working.
We are, so the lecture slides will be uploaded as we go, right? Because there will be adjustments as we realize where you all are. But they'll be at that link. And then there's two things we'd like you to vote on. One is, what do you want that last lecture to be? And I think maybe we should wait a little, wait until tomorrow afternoon so as you have a sense of the things we're doing. And then there's a couple of options. I can do a lecture on you know, like lab leaks, or we can do one on One Health, or we can go through some statistics, or we can try and build a model together to tackle an evolutionary question. You know, so I just, you know, give it, or if there's something that you think that we're missing that you really want to do, talk to us and we can add it to the list of options. Yeah? And then we are running Journal Club on Tuesday, on Friday. And so again, we're giving you options. So give us a sense. What are the options, Ben?
There's the last slide on this, but I think there's sort of like malaria and more vector-borne diseases. There's seasonal dynamics and directly transmitted infections. There is pathogen host arms races and coevolution. And the last one is the evolution of virulence, I believe. Don't quote me on that, check the last slide.
This is so perfectly prepared.
There's 4 options, and then based on what people have interest in, we can select a paper and then send that paper out, and then we'll discuss the paper. Given the diversity of backgrounds, don't feel shy or uncomfortable if you don't have a strong background and don't understand all of the methodology in that paper. We'll try to make the discussion approachable for people from the physics to the oceanography background.
Mostly it's about oceanography, I think, right? Okay, so let's get going. I think to understand health, you need to understand a vast array of things. And a lot of what we're going to talk about today is kind of the detective work, which many of you are familiar with, as half of you have worked on COVID, it seems, that you need to actually figure out what burdens are and how they're shaped through time. And the way I've sort of organized my thinking about this is you can think of sort of 5 levels of things that matter. The first is measurement. The second is how you evaluate associations. This is the very classic of, you know, smoking causes lung cancer. Microbial ecology is inherently different because it's as if smoking causes smoking, which it might, right? There can be social transmission networks. But that leads to inherently different mathematics, inherently different dynamics. By the way, my accent is strange. I talk really fast. Please just go if you feel I'm talking too fast, and I'll get it. Or Ben, you tell me. So an evolution obviously matters. If you're a biologist, it will come as no surprise that evolution of antibiotic resistance is one of the vast, you know, scourges of the current time that we need to be grappling with. And of course, the ways in which the world works will shape how evolutionary processes happen. So thinking about that also contributes to thinking about health. What I'm going to do today is just give you a quick overview of all of these different pieces, and then tomorrow we'll start digging in a bit more. And this will also give me a sense of when I see you yawning, I'll be like, okay, a little faster, or if I see you going, we'll do a regroup. And Ben will be monitoring facial expressions. Okay, so measurement sounds really boring, and it, you know, measurement of populations, measurement of things. But it turns out it's really important. A very simple example, if you have a population of 1,000 individuals, half of them have a disease. In almost every system in the world, you're not counting everybody who has the disease. Nobody has a surveillance system that good. And so to even understand what the distribution of diseases in populations, you need to have a kind of model of how the world works. And the model in this case is a model of reporting, right? You need to know that we're only getting 1 in 5 of the cases reported. And if you put those 2 things together, this model of how measurement works, you would end up with a sensible estimate. This seems like a really trivial point, but I think it underscores the fact that in any situation in the world, we need to think about what it is we're seeing and how it is that that works. A very classic example of a sort of related issue that many of you probably know about is this issue of that was encountered by the statistician Abraham Walt. And in the First World War, that's an error. Ben, why don't you edit that? It's my mistake, it's not Ben's mistake. So he was asked to try and get to grips with how to minimize bomber losses that would sustain damage due to enemy fire. Okay? So this was the data he got, which is the planes that came back that had these bullet holes in them, the red dots are bullet holes. And so then he had to decide where to put the armor, where to reinforce the planes. So what do you think he concluded? Some of you probably know this. Where should you put the armor? Yes?
Where it's not the red dots.
Exactly. Do you want to explain why?
Because if they don't record, it's because they was hitting them.
Does that make sense to everybody? Yeah, everybody knows this example because it's a good one. But the problem is that you have inherently the thing that you're measuring contains information about what it is, right? So again, you need a model of how measurement works. You need an understanding of how the world works to get this right. So you should be putting armor as, tell me your name again. David? As David said, you need to put the armor exactly where there aren't red dots, right? Which, it seems obvious once you say it, but at the time it was quite the breakthrough. And this is an example of survivorship bias. It's another one of those pieces you have to think about when you think about measurement, when you think about getting to grips with what your numbers are actually telling you. So you have a sort of meta-modelment of how measurement works. And as perhaps an illustration of what just happened, there's a fantastic XKCD cartoon, which is like, raise your hand if you're familiar with selection bias. As you can see, it's a term most people know at a statistics conference. So, selection bias. So the basic point I just wanted to remind us of and put us all together with, and again, so many of you who worked on COVID, this is probably unbelievably familiar to you. Counting things is really hard. Counting things turns out to be actually one of the major challenges in epidemiology. We cannot find all the people who are sick. We don't know what the denominator is. We don't know who is at risk of getting infected. It's hard. It's weirdly difficult. It's incredibly important. It's strangely boring, so we're not going to talk about it much more. But I just wanted to remind us that this is where it all starts. A vast amount of the rest of epidemiology is founded on evaluating associations. And so this is an example from this website here, which shows you divorce rate in Maine in the red line, and the per capita consumption of margarine in the black line. And so margarine is a butter substitute, just in case, yeah. I didn't know what it was. And so, then you might, if you're looking at that association, you'd be like, well, clearly margarine causes divorce. So this would be your kind of hypothesized model of what's going on. Your exposure is margarine, your outcome is divorce. You might be thinking, how could that possibly happen? Well, in this instance, it's obvious that probably what's happened is actually time. I mean, first of all, you might try and think, oh, does margarine cause divorce? Does it make people grumpy so they get divorced more? Is there something about, you'd want to come up with a mechanism, even for a really crazy example like this. But in actual fact, probably what's happening is that so many things change with time, and we have time inherent in this relationship, right? The plot actually has time on the x-axis here. So if you think about, you suddenly bring time in, which is this hidden variable, then perception of health associations of margarine might lead to that decline. There might be more other food options available, and people have suggested many reasons as to why the divorce rate in Maine fell, one of which is that people wait longer before they get married. So they're likely to pick someone they're more likely to stay married to. This is when I moved to America. I have to say, everyone's getting married so young. It was really freaky. We British people don't do that. And so the issue here is one of confounding, right? So there's a hidden variable that's driving that relationship that unless you account for it, you're not going to get the right thing. And there's a little snippet of code that goes through an example of that. And that one way you can tackle this is by drawing exactly this sort of graph. They're referred to as directed acyclical graphs, which allow you to formalize what your assumptions are about the relationships, right? And you can never know. This is something that's sort of fundamental in epidemiology, which is that we're always building models of how the world works. What we're trying to do is be as honest and explicit about our assumptions as possible. You can't know for sure, but you can make assumptions about what the things that might matter are. Another example of where things go wrong so this is where you were missing that third variable. Another example of where this might be the case is based on a classic example from, I think it was Berkeley. It's Berkeley. So they looked at the population of male students and female students that had applied, so the full bars are the applied and the darker bars are the accepted. And it looked like more males were being accepted than females. And this was worrying. This was, what, the 80s? the 80s in Berkeley, and people are like, well, this is not acceptable. You know, why are we accepting more males than females? What's going on? Can anyone think about what might have been going on? Again, somebody might know this example. This is a classic epidemiology example. Yeah?
Well, I know the example, and it was like women applied for more difficult majors than acceptance rates small.
Brilliant. Clearly, I can turn over teaching the class. So, did everyone get that explanation? Okay. Okay. Oh, yeah. Do you want to do it again?
Okay. The matter was that women were applying to different majors than men, and they were applying more to the majors that had inherently a smaller acceptance rate. So that's why they were getting in less.
It's a perfect explanation. I should just choose examples that everyone else can explain because it's much easier. Tell me your name. Talis said there is a missing variable in this analysis, and that is department that they applied to. And if you break it down, so this is entirely simulated data. I just made it up. There's some R code that gives you this in that Dropbox folder. But if you ranked departments by how much they accepted, what you found was that females were applying to these much more difficult departments. And if you accounted for that, if you put that into the model, classic regression model, logistic regression in this case, you find that there's no difference. There's no sex effect on acceptance. Okay? So you would need to expand. You're looking for the association. Our question is there an association between exposure being sex in this case and acceptance rate? And you need to control for department before you do that in an appropriate way. Controlling for a third variable is a sort of huge bedrock of what we do in epidemiology, in this classic epidemiology. And you use it to adjust for confounding, so things like time or things like department. But you might also care about what's happening. It might be that, you know, my risk of the incidence of chronic disease might be different in males and females depending on whether they had had some early infection or not. So you might build a model that had all those pieces in it, not because you wanted to control for nonsense, but because you cared about the biology of that piece. Okay? There is you might think, okay, then shouldn't we just put everything in because it'll make everything better? It turns out, no, that would be too easy. As an example, one could evaluate an association between, you know, you might think clearly people who are methodologically crap, rubbish, bad, are making very bold claims in science, right? It's like, I'm fast and loose, I'm a bit of a cowboy, I'm like, I'm going to move that regression variable over. Therefore, I have discovered the cure for cancer, right? So you might think that this was going on if you were feeling dark and cynical about the way science works, which is perhaps not inappropriate in some instances. So you would want to associate this relationship. And maybe you find a sort of cloud of points like this, which suggests there isn't much of a relationship. Maybe there's a hint of a decline, but it's really not there. But you become concerned about the fact, you're like, well, maybe publication bias matters in this instance, right? Maybe what makes it into the publication literature is different from what doesn't. So I'm including studies from both the published and unpublished literature, and I'm going to include publication as a third variable. Okay? And if you do that, you actually bias what your inference is, because it turns out to be published at all, it's a little bit like the example that Talis mentioned of the airplanes that come back. Actually, you didn't mention that. David mentioned that. The things that are published are likely to be really bold, right? Nobody publishes a really boring result. And the things that are published are also likely to be at least somewhat methodologically rigorous. So just by this selection process, this is kind of a survivorship bias, you end up with a result associated with, a negative result associated with including this third variable, which is publication. So you're getting it wrong by including this variable overall. So you're introducing a spurious association, and it's called a collider, for those of you who have done this sort of thing. All these things, again, all these things are kind of, for those of you who are more mathematically oriented, these may seem exceptionally boring because they are sort of three variable problems, but they're unbelievably important, right? And I'm sure you've all read headlines in epidemiology that sort of go like, smoking can cause hypothermia in men from 25 to 40, right? It really feels like it's just a kind of like, pick the thing. And this is not what we want. And a lot of these problems, I'm sure many of you have heard about the replication crisis as well. It's like when you try and repeat studies that have been done before, it proves to be impossible. And there are many reasons associated with this, but thinking carefully about what you're measuring and then thinking carefully about what associations you're testing is a huge part of this. And even if the association you're testing, even if the thing you identify is wrong, which it can be, like science is all about getting things wrong, if you have been explicit about your assumptions, it should be possible to recover that error. If you haven't been explicit, it becomes much harder. And one way to be explicit is to very carefully write down the diagram of all the flows you think are happening. So this sort of quasi-mathematical, this mathematical adjacent language is just a way of being very clear about what we think is going on. Okay? So this is just a downer of a set of results. Counting things is really hard. Bias and confounding is ubiquitous. These are the two sort of, I think, bits around the foundations of things we just have to keep in mind as we move forward in epidemiology. For the rest of the course, that was very much what you're going to get on that, unless you want to do more. We can revisit if this is of interest. For the rest of the course, so we're going to start by, I'm going to reintroduce a little bit of microbial ecology and evolution today, and then we'll dig into these more in the next couple of days. Microbial ecology, you move into a different space, because there we were thinking of my exposure, smoking causes my outcome, lung cancer. For something like measles infection, which is something I work on a lot, the exposure, measles, is the outcome, measles. So you have a feedback loop. And the moment you have a feedback loop, you're in a different world, right? You're no longer in a directed acyclical graph, you precisely have cycles. So you have to account for those cycles. And there is vast motivation for doing this. It feels silly saying this to a room, half of whom have worked on COVID, but obviously we care about digging into how infectious diseases are spreading across landscapes, trying to come up with the tools to understand what might be going on. Ronald Ross, who got the Nobel Prize for discoveries around malaria, used the term dependent happenings to capture this, your exposure is your outcome. So the way you would write that down is, so for example, for something like measles, what we do is we assume that everybody in the world can be put into one of three boxes. You can be susceptible, you can be infected, or you can be recovered. So we put everybody in the world in these boxes, and then we have to describe how individuals are moving between the boxes, right? What are the rates that are moving people between these different groups? So in this case, to move from S to I, obviously transmission has to happen. And since transmission happens from infected individuals, we have this little dashed line that is capturing that dependence, the dependent happening. And once you're infected, at some rates you recover. Individuals enter the susceptible class via births. They're just raining from the sky in this example. And then in all instances, one can die in this particular example. Okay, so you could put parameters on this. Mu is often used for death. Beta, the transmission rate, and you multiply it by I for where it's coming from, and then recovery is often captured by gamma. Okay, let's write down the equations for this. You can either tell me what to write, or someone can come up and do it. Or I'll make Ben do it and he'll kill me. David. Should we chant? David, David.
Have you explained what is a term?
Yes, please. I'd love to know.
Yes.
Okay, so we have the bird. There is the total population. There is N. We have the normal death. So we have mu multiplied by death because this is outside this box, and every baby born like susceptible. So we have to put here the term of interactions. So we say that the susceptibles and infected people are fighting each other randomly in the space. And I think that that's the only equation that we have. So we have the other. So now the susceptible becomes infected, so this is positive. And we have the normal rate, the same, so this is I. And we have the recovery time, so we have the number of days that we have to spend to have another, to recover from the infection. And where is the, so, and at the end, the RDT, so the infected people becomes recovered people, and finally they die, sorry, they die, and I think, yes, finally, yes, because that's the end of the, yes. And I think that is that, right? Because we don't have, yes.
That's perfect.
Sorry
for
the...
Thanks, David. Wonderful. So I was told that you had done this before, so I'm hoping that it's mostly a little bit of revision, a little bit of putting it back in your brains as we go forwards. So I'll do a little bit of like asking you to follow it through. We can make sure that I got it right by comparing to, looks like we're in agreement, that's good. To add vaccination, I don't think anyone needs to come up, but imagine we had vaccination at birth. What would that look like? Yes? Tell me your name.
I'm Ruth.
Ruth.
Yes, I think that we need to add other compartment.
Ooh, okay.
And we have to remove the infected people to vaccinated people.
That's a one.
No, sorry, to susceptible. So susceptible to vaccinated.
Great, great. That's exactly right. I think one way that I was going to do it in this, what you proposed is correct. I was going to add vaccination at birth. So instead of having the arrow come from the susceptibles into the vaccination, I was having them directly removed there. But I think it's exactly the same principle, right? And I think everyone can see where that would go in here. You know, we're looking at one minus, oh, I have it up there anyway. Yes? Yes? Okay, isn't it nice doing things we can do? So let's imagine, so this was a situation where we have births coming in, we have deaths going out. Let's imagine we have a closed population situation. This is where nobody's being born and nobody's dying. So what's going to happen? Let's imagine we start off here. Everybody's susceptible to begin with, and you drop one infected in, and the R naught is, say, 5. What's going to happen next? Say that again? So the susceptibles are going to fall. What are the infecteds going to do? But all the way, or?
Stop.
Uh-huh.
Plateau.
People are waiting. Tell me your name again as well, please, sorry. That's good. It's going to be hard to remember.
I said it. My name is João. The infected ones will increase until they hit a plateau when everybody's kind of affected, but they cannot infect anyone else because they're not births or death.
Mm-hmm. Yeah, perfect.
But in this dynamics, we are not accounting for some type of immunity, so they will just remain infected.
So I think we actually have, if you're infected, you move out of the infected class, right, and into the recovered class.
Yeah, but I thought that there was an arrow going to the recoverer to the susceptible one, so like, oh no, that's the opposite. Yeah, so.
So what's going to happen? Tell me your name again, sorry, I didn't get
João.
it. João, okay. So the susceptibles are going to fall, that's clear. And as they fall, the infecteds are going to come up. And then what's going to happen? So the recovereds are also coming up. This is susceptibles, this is infecteds. The recovereds are coming up as well. It's going to go down. Yes, exactly. Does that make sense to everybody? So you're having flow into the recovered box from the infecteds, which means you're having flow out of the infected box. And this is a really magical point, which is one we're going to return to often. Yes? Okay. So that's what that looks like in colors. And this captures one of the key elements of infectious disease dynamics, which is that if you start off, so here the red person is infected and the R0 is 5. So the number of new infections per infected individual is 5. And as you move through time, more and more individuals are recovered. So in some sense, there comes a point where this infected individual is wasting 4 of their contacts. As David said, we're assuming they're in contact at random with individuals in the population. They're using up 4 of these on transmitting to individuals who are already recovered. So they're not infecting 5 individuals anymore, which means that it's going to collapse down, as Zhao mentioned, to eventually there will be no more infecteds. In this instance, you have to run out of infected individuals. The infection will be eliminated. Yeah? Make sense? So what would happen if there were births in the population? I've basically told you the answer. What happens to infecteds at the end? Would you have, so, Thales? So you're right, but that's more complicated than what I was looking for. So in this case, the infecteds go to 0. If there are births, then infecteds can go to an equilibrium that's above 0. And there is a little bit of cycling on the way, but it flattens as Zhao. Yes. Does that? Yes. Good. Okay. So this is, the very 1,000-mile introduction to microbial ecology, and we will do a lot more of this and a lot more of trying to bring this to grapple headlong with data in the subsequent lectures. Okay, so the last piece, we've talked about measurement, we've talked about evaluating associations, we've introduced how microbial ecology brings you into a different space, a different world from classic epidemiology. And now I am going to swing into a brief intro to how evolution works in this setting, which will be abundantly familiar to many of you, but perhaps less familiar to some. So evolution describes the overall relationship, the fact that inheritance by descent is something that characterizes our species and every other species on the planet. So this one way in which we represent this is by phylogenies. This is something Ben knows much more about than I do, so I will be directing questions to Ben. We hominids are related, back to the base of the evolutionary tree to various other primates. You can write down exactly the same sort of relationship for coronaviruses. So on this map, we've shown the 4 endemic coronaviruses that circulate in our populations regularly. And the red ones are the emergent coronaviruses. And they tend to have different hosts, but there was a point at which they diverged from each other and entered into new hosts and spread, et cetera, et cetera. Same principle as for us. And then, of course, there is, the example of influenza. This is influenza A, and it shows you a very spectacular structure of this evolutionary tree, which captures something very basic about how the evolution of influenza works, which is that it seems like pretty much every year, every strain except one goes extinct, right? So there's complete replacement. None of these branches are reaching out to here, they're just gone, right? So it'd be like, as if we had spread through the planet, had eliminated all the other hominids. I realize as I said that's a terrible example because that's kind of what we're doing. Anyway, moving on. We think this is to do absolutely with how immunity works. So what happens is that an influenza virus spreads within the population. We all acquire immunity to that particular strain. There's a little bit of evolution. One manages to escape and spread in the new population. Okay. million mile view of how evolution works. The four forces that people usually consider when they're talking about evolution is mutation, migration, selection, and drift. And so mutation, it sounds like none of you need an introduction to this, but it's kind of like spelling mistakes, right? You have a gene, things get miscopied. Most of these miscopies end up in really bad outcomes for the whatever it is that we're considering, but some of them will end up being good. These will spread in the population. But that's not, mutation is really just about the mistakes. So when people were saying things like, oh my God, SARS-CoV-2 has such a mutation rate, it's like everything has a mutation rate, right? Everything mutates all the time. We're mutating. Let's be calm. It's not the mutation rate that matters, it's what the mutation turns into. Migration is another way of moving genes around, same basic principle, but the two things that sort of people fixate on a lot are both selection and drift. A quick primer on selection, forgive me for the baby style, but I think it's always useful to just start at the beginning. If we imagine we have an initial population where every individual, these are the parents, these are the offspring, have a single offspring, and then there's a mutation, which ends up resulting in the offspring number being doubled, then what we expect to happen, so these things are living 1 year, producing one offspring, and then dying because life is rough in my made-up world. The next generation, this guy's going to have 2 offspring where everybody else just has 1, and his 2 offspring are going to have 2 offspring. I don't know what, her 2 offspring, much more likely, and so forth, right? So through the generations, in a very short amount of time, if you have double the offspring of someone else, you will end up completely dominating the population. This is selection. Any mutation that alters survival or fertility will result in a change in selection, could result in this sort of process. And any gene that doesn't affect survival or mortality doesn't, right? It's, if something changed the color of your eyes but it did nothing to your reproductive success, nothing would happen. So it really does need to shape your demography. Classically, fitness is usually defined using a range of different measures, but here's one example. If you map out proportions surviving over age and fertility over age, so survivorship is, we start with 100% surviving at age 1, say 50 at age 2. It's always declining, right? It has to decline. That's how survival works. And then fertility, in this made-up organism increases like that. You can express fitness as the number is sort of very similar to the concept for infectious diseases, right? It's the number of offspring on average per reproductive female. Okay, so who can tell me what the fitness would have been for that previous goofy example I showed you for this thing? Yes, exactly right. Tell me your name? João, okay. João, okay. Can we all go by João? Exactly right. So the mutant they survive 1 year. I told you they all survive 1 year, and then they die, and they produce 2 offspring. That means their fitness is 2. The ancestral state has a fitness of 1, so clearly the one with the highest fitness wins. Exactly right. Selection is half of the story. The other half is drift. And if you were to introduce a completely neutral mutation into a population, so it has no effect on fitness, what's going to happen? Say we introduce it right here. What do we think is going to happen in terms of its frequency within the population? Yes, the person next to Talis. And your name, perhaps. João?
Not also. I am Cauê.
Cauê.
Well, many things may happen. That if you consider neutral theory, that mutation may fixate, which means would happen as the same as the red mutation we had before, but it may also go away because it has no effect on fitness. So it would be completely subject to randomness and those effects.
This is exactly the 2 bounds Cauê described are the only fates that can happen, but also, as you said, many things can happen. And this has been one way in which people have made an analogy is in the long run, a completely neutral mutation, if nothing else happens, will either be extinct or it will be at fixation, it means that everyone in the population has it. And this is then described using the drunken walk analogy. If this is like zero, like extinction, and this is complete fixation, you can imagine the mutation is starting here, and it's taking one drunk walk, and one drunk walk, and eventually it's going to hit one of the edges, right? That's how one can imagine this. Not that I know. Okay, so one example I think is very helpful for thinking about, I think it's really important that we always remember that evolution is both selection and drift. It's exactly when people forget drift that some of the weirder sides of evolutionary biology come out. A really good example of this is when we think about the evolution of senescence. And this is the fact that as we grow older, our risk of dying increases, right? And we also get more fragile, and like I get tired more than you guys, I have more wrinkles. Senescence seems to be ubiquitous across species on the planet, right? It's something that happens. So why would this happen? If you're thinking of fitness in really basic terms, this seems kind of weird, right? Like, surely everything should be reproducing as much as possible, surviving as long as possible. Why do you have this consistent decline in functioning with age? So this was sort of figured out by Medawar and Williams and some of the great giants in evolutionary biology using the simple example. So if you imagine you're in a population where, so let's just focus on a cohort where 2,000 individuals are born, and the probability of dying is 50%. By age 2, there's 1,000 individuals. By age 3, there's 500. By age 4, there's 250, and so on. So, no matter, even if mortality is completely constant across age, you end up with many young individuals and very few old individuals, right? And what that means is, so if we put down our probability of dying as being this, if you imagine that there's a mutation that pops up that increases mortality at a young age, it's going to considerably decrease the size of the population, and thus you can imagine it's going to considerably decrease fitness. However, if instead a mutation pops up that increases mortality at a late age, there's basically nobody there. So there's no discernible effect, right? And so what that means is that any mutation that pops up that increases mortality at age 110 in us is just going to spread to fixation, right? Like there's nothing to stop it. No one ever expresses it. Just by chance, it's going to end up getting to 100%. If there's something that pops up that increases mortality at 45, there might be sufficiently few individuals that survive that late that it will spread in at least some individuals. It's expressed rarely. It will also go to fixation. So we expect mutations that increase late life mortality to accumulate, right? Which is why senescence is inevitable, unfortunately. Both processes, both selection and drift, might underpin this. I've been describing essentially drift, which is where the mutation pops up and there's nothing to stop it. So it just spreads in the population because it's just sort of so rarely expressed, there's nothing to hold it back. But you also could have antagonistic pleiotropy. And this is where, for example, something about some investment in bone strength when you're young reduces the resources you have available to survive later. So trade-offs, basically. You're investing in young individuals at the expense of old individuals. This is selection. Okay? So the evolution of senescence contains, I think, both of these really important ideas, the side that is associated with drift and the side that is associated with fitness maximization. And fitness maximization gets a weird, it can move you into very strange terrain when people are like, oh, survival of the fittest, like evolutionary psychology, psychotic stuff. But I think which is one of the reasons it's so important to be clear about how we think about these things. Okay, how does evolution intersect with health? We're here to talk about health and epidemiology, and clearly in lots of different ways. It can shape the evolution of pathogens, so how fast they spread through populations, as anyone who worked on SARS-CoV-2 probably knows far too intimately. It can increase virulence of pathogens, and we'll talk a little bit about that in the third lecture. It can shape immune escape, so it can give you that sort of influenza tree, or there can be a lot on resistance to therapeutic treatments. However, we can't just think about pathogens, of course. We need to think, it's really interesting to think about also our own evolutionary history and what that's done to our health. There are many genetic diseases that are sort of underpinned by our history of infection by pathogens. A classic example is sickle cell anemia, where individuals who carry a particular mutation that changes the shape of their red blood cells are resistant to malaria, but can suffer from like very debilitating clogging of red blood, their blood cell pathways, etc. It can be really, really devastating. And understanding that health outcome doesn't make any sense unless you know about the malaria background. Treatment effects are very different often in different contexts. A really important and weird part about thinking about infectious diseases and health is that the thing that kills you almost all the time is your own immune system, right? Like COVID-19, people were dying long after they cleared the infection. And this has got to do with, having an immune system is like having a really dangerous line of defense. It's like having a militia run your internal defense program, and occasionally it goes crazy. And this probably, the reason that militia exists is because of our history of evolution of infectious diseases. And understanding that, again, can be important. This is something that Ben and I, is that fair? Feel a little ambivalent about. But people often think a lot about the ways in which our current lifestyles are at odds with how we'd lived historically, sort of paleo-historically. So for example, the epidemic of obesity might be partly because we're maladapted to the diets that we have now. I think the reasons that we're hesitant about this is it's so hard to know exactly what your exposures would be in the past, and it's so often it so often links to fuzzy arguments about exactly what's going on. Not that one would dismiss it entirely, but it's worth thinking about what you're assuming about the past before you move forwards with these framings. Okay, and then of course, the tricky thing, the bit that underpins all of this is, I told you that pathogen dynamics has these feedbacks in it. Of course, if you start thinking about pathogen dynamics with evolution, with us, you have all sorts of feedback dynamics, because we've changed because of the way they change, and they change because of how we change, and it's just a whole big mess. Okay. So what I want you to remember is that counting things is hard. Bias and confounding are ubiquitous. If you're thinking about things with these feedback cycles, you need to start thinking about cyclical graphs where suddenly outcomes are harder to predict. Evolution is both chance and selection. And we might talk a bit about challenges, but it will depend what you guys want to do. The way this course is going to work, again, just another overview, is in this introduction, I've given you all I'm planning to give you on measurement and evaluating associations. We can go deeper down that path if this attracts some of you, but we don't have to. Our plan for the next 2 lectures is to talk a little bit more about microbial ecology. So go deeper into immunizing infections and start probing how you move from these extremely platonic relationships to actually marrying the data with what's going on. And again, snippets of code throughout that will link to what we're doing. We will also talk about vector-borne infections. So what happens when you have a mosquito in the mix, and again, what happens when you try and take your mosquito human data and map it to these really simple models. And then we will talk a bit about pathogen evolution. And so the 5th lecture, again, I'm leaving it open. I want to get a sense of what you want to know, what you're interested in. Do come and talk to us. I think maybe we'll have some open chat now, any comments and questions you might have. Because, yeah, I don't know what to tell you. And I'd like you to tell me what you'd most like to know from what we've been doing. I think that's it. Okay, and these are the options. Oh, thank you, Ben. So there's the link, and these are the options that we proposed. We could talk about using big data, so social media data or mobile phone call data records to get to grips with epidemiology. We could talk about lab accidents and bioterrorism. The students at Princeton love that. You guys don't have to love that. We could talk about One Health. It's a sort of where we start, the intersection between humans and diseases, and we start sort of triangulating on that. Ben, E, and I are just starting a research project on that, so it's one of the things we're doing. We could just spend some time going through R code, sort of grappling with the snippets of R code and stuff. Or we could develop a model to tackle an evolutionary question together. So for example, something that has puzzled me for a very long time, I don't have the answer, so I'm hoping we could come up with something together, is the fact that your immune system is made up of 2 parts, innate immunity and adaptive immunity. So innate immunity looks for conserved signals of pathogens. It always does the same thing. It sees encoded receptors, it goes for it. Adaptive immunity is the bit that learns. And for some reason, over the course of senescence, adaptive immunity falls apart first. So adaptive immunity senesces faster. Why would that be? And it might be something to do with how the costs are paid. It seems like you build your adaptive immune system really early in life, whereas your innate immune system, you might be paying the costs all the way through life. So we could try and build a model to probe what that would mean and how we would interface that with infectious diseases over time. This is a possibility. It's one of the things you can vote for. I don't know how that will go because, as I said, I don't know the answer. It could be really hard. Another option is to develop a statistical model to tackle a public health issue together. So for example, Ben and I work a lot with the demographic health survey data, which is these vast cross-sectional surveys where they ask, mothers, has your child had a fever? Has your child been vaccinated? Does your child sleep under a bed net? And so you can start building maps of what these covariates look like, how they've changed over time, how they link to other processes. We have tons of data which we could work with together to think about these questions. I think I also circulated a PDF of some of the other publicly available data sources that you might have ideas that you wanted to tackle together. And for these last two, we could, of course, split up, where half the class did one and half the class did the other. If the voting falls out that way, and talk amongst yourselves as well, we could go in that direction. So, and then this, Ben, you have shared that, right? The link for the...
Yeah, this is in the slides, and the link is on the Slack. I would suggest, so please vote for your preference by Tuesday. You can maybe wait until the end of the lecture on Tuesday to get a better sense of what we're talking about, and that may change your opinion, but feel free to vote when you see fit. And feel free to propose things outside these options as well, whether for Journal Club or the 5th lecture on Friday.
And we'll stick around for a while, but otherwise, that's what we got for you.
I might suggest downloading the article snippets. I might suggest trying to download the R code snippets now and just making sure they work. 'Cause I know sometimes there's package issues and version issues. And I'll stick around and just raise your hand if something's not working, or you can't even download the things. So I might suggest doing that now with the time we have remaining, but also we're around for questions.
And suggestions.
And suggestions.
Yeah. Thanks, Ben, for sounding calm and collected. It's something I enjoy very much. But I think we're getting our answer right. They do really know the answer.
Can you please remind us what is One Health? Yes.
Yeah, there was a question about One Health. Sorry to interrupt, Jess. So One Health is a concept that's applied to a way of thinking about the way human health is determined. And One Health is a new term applied to an old concept. But it's basically about, you can think of a triangle where you have human health, animal health, and environmental health all connected. So things like zoonotic diseases that occur because you clear a forest and there's contact with the viral reservoir in that forest causing an outbreak in humans. Another thing you can think about is domestic animals bring diseases and then contact wild animals and then give the diseases to the wild animals. That spills over. And it's mostly focused on this relationship between human health and environmental and ecological contexts. And people are increasingly interested in this, and including me and Jess. Yeah.
I just realized, talking to Isabel, that I had failed to explain the senescence process very well. And we're not talking about mutations that happen in any part of your body, right? Like, I probably have more mutations in my skin than you guys do because I'm older, but they won't get passed on to my offspring at any stage. What matters is mutations that happen in the germline. So if I was to develop a mutation in my eggs that resulted in an increase in mortality at age 70, that meant that I had a risk of dying from heart disease at age 70, that could still spread in the population because I would have kids, well, I wouldn't actually, but I might have had kids all the way up to age 70 that I would have passed on to the next generation. Thank you, Isabel.
Oh, okay, okay. So we'll upload them and then I guess you'll change the path to
Yeah, just that we don't have the data.
May I make an announcement about the snippets? So it seems like you all have the snippets working fine. You'll find out where they're relevant at the bottom of the slides. I don't want to force anyone to go through this if they don't. They're also full of errors, as Mateus just pointed out to me. So do flag the errors when you find them. But it really is to give you the option. We're going to be talking about these equations and patterns that gives you the option of taking them into R. There is no order in them. It seems like you all have access to them now. That's great. No worries about... Ben and I teach a lot in Madagascar, where like everybody's computer is broken all of the time. So we're quite used to spending ages going like, do you have R? Do you know what R is? But yes, we seem to be ahead. So yeah, just general questions then, general chat. I think you all have proven yourselves more than proficient with the R codes, and we will bring them up as comes along. If there are no more questions, I mean, we will be here for 5 more minutes, but then afterwards I would encourage you to go and enjoy the day. Not mentioning the